All right, so listening up. So for today's do now question, I want us to throw back and think a bit about the structure and bonding. Remember, we covered two types of solids for the structure and bonding before we paused and started our internals, and that was the metallic and the ionic. I want to talk about ionic because currently we are looking at precipitation reactions, and so we need to think about why some ionic substances are soluble in water and why some are not. So that's what I kind of want to talk about today. So just as a reminder, when we talked about particles that were soluble in water, plus, minus, plus, I'm going to actually do it nice colors, minus, do 3 rows, plus, minus, 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 minus. All right, so this is my ionic solid. Agree? Ionic solid? Yep. So if the ionic solid is soluble in water, so here's my ionic solid. I feel really thrown off because I'm not writing with my normal pens. All right, so here's an ionic solid, and this ionic solid is soluble. So what that means is that a water molecule can come around. Now I gotta remember which way I want. Like that. Here's my oxygen and my hydrogens, the slightly negative and the slightly positive. So when I have a soluble substance, the water molecule can go next to my ionic solid and start interacting with those ions, and remember, they can pull those ions out. So if I had, say, for example, a positive ion, it would be surrounded by the water molecules like that. Whereas that is slightly negative and that's slightly positive, so that goes out here. And then for this guy over here, you would see the negative ion like that. Slightly positive, slightly negative. So that's what happens when it's soluble, remember? All right, if it is insoluble, I'm gonna grab some more colors. So you know me, I'm a color fiend. I'm trying to get colors that will actually be new colors. All right. Let's say I have something that's insoluble. So I would have all those positive and negatives. Headphones? Yeah. This one is another ionic solid, insoluble. Here's another ionic solid that's insoluble. Let me just put in my negative ions. All right, do you see how there's another ionic solid? Agreed? Now this one here is an example of an insoluble one. So the water molecule will go towards the ionic solid as it normally tries to, but in this case it will not be able to pull it apart. So if it cannot pull it apart, then it's insoluble. If it can, then it's soluble. Are we okay with that so far? So can isolate ions, it's soluble. Insoluble is cannot. When we're thinking about why some things are soluble and why some things are not soluble, we have to ask ourselves, is the ionic compound more attracted to itself or more attracted to the water molecule? In this example where it is soluble, the ions are more attracted to water than itself. So itself versus water, it's greater attraction to that water. Are we okay so far with that? Yep. If it is not soluble, then the ions are more attracted to itself than the water. So itself greater than water when it comes to those forces of attraction. So that is why some things are soluble and some things are not soluble. Now this is going to apply to two standards that we are doing. The first standard, now I'm trying to look for some more colors. This is really hard to kind of deal with all these colors at once. All right. Some things to keep in mind when we are dealing with solids. When we're dealing with these two standards. When we are talking about structure and bonding, they are most likely going to ask you about a soluble ionic solid. All right. So structure and bonding external, most likely they will give you a water soluble ionic solid. Most likely. I haven't seen an insoluble one. I have seen them compare soluble and insoluble ions before, but I think that was on a mock exam, which tend to be harder anyway. So I have seen them ask this question where they ask you to compare and contrast a soluble versus insoluble ionic solid. But like I said, I don't think I saw it on an NZQA assessment. I think I just saw it on a mock exam many, many years ago, because it is quite a hard question. So most likely assume that your ionic solids are soluble in water in the context of the structure and bonding external that we were doing before this standard. In this standard, however, when we are talking about soluble and insoluble ionic solids, the insoluble one is the one we're referring to whenever we talk about precipitation reactions. So in our ion identification, insoluble ionic solids are the PPT, the precipitate. So what happens when we are mixing our two solutions together is we are mixing two soluble solutions. Both of these soluble solutions have ions floating around. Now, when I mix those two solutions together, the new ions will interact with each other, and potentially, with the new ions interacting with each other, you are going to get a combination that will have a greater attraction to itself than to the water, making it insoluble, making it the precipitate form when you mix those two solutions. If you didn't mix those two solutions together, they would have stayed in solution. So it's just that specific combo. Don't worry about explaining this. We're never going to ask you to explain this on the, We're never going to ask you to explain this on the ion identification internal that we're working on right now. It's just to give you background context. It might, in a very rare situation, come up on the external, but I highly doubt it. But it's good to see it, and it's also good for just comprehensive understanding of the chemistry material. Are we okay with that so far? All right. I also wanted to explain that to you because I think it's good when you guys do the external to kind of give another layer of understanding for the solubility property. All right, moving on. Okay, so I'll pause the recording for now because we're going to stop the notes and I'll give you the intro for the lesson. All right, so I have a beaker here. And this beaker has a blue solution in there. Now, we've done some experiments before, so we might have an idea of what this could be. So before we even do our flowchart tests, what do we think this might be? Copper nitrate, why? Because it's blue. Because it's blue? So we've learned that blue is often, now is the blue associated with the copper or the nitrate? Copper. The copper. Because if you guys remember, you guys were given other things that had nitrate in it. That wasn't a blue color. Could this be copper sulfate? Yes. Yes, it could, because again, we're talking about the copper ion again. So we know the copper 2 is a blue color. It could also be copper sulfate. So what you would do in this qualitative analysis is you would figure out what's there. All right? Yeah. It is not actually copper sulfate or copper nitrate. The face, you guys were really scared. This is just Powerade. I love pulling that prank on you guys because obviously you're very serious. You're like, oh yeah, it could be copper, and then I just take a swig. No, this is just Powerade. And if you guys want me to prove it, I can. We can get the solutions out, and it's not going to react the same way. Let me show you. So, it is just Powerade I brought from home to prove a point. Let's not drink things, obviously, in the chemistry lab. I just did that as a fun little joke. Let me prove that it's not that. What are you doing? All right. So, if you guys remember from your flow chart, all right, so blue solution, right? If you guys remember from your flow chart, if you mixed in sodium hydroxide, you should get a blue precipitate, correct? Anything happening? No, not copper sulfate. Or, no, I'm not going to drink it now, because now I've actually added sodium hydroxide to it. But just trying to teach you guys about some of the science. I'm also going to check this one out now, because I'm super paranoid. Huh? No? You don't want me to do that again? Wait. All right. Do what I want. Okay, so I'm just trying to think what else I had planned for this lesson before we go on to it. All right, cool. So, I'll pause the recording. We don't need this anymore. Actually, yes, we should, because this is actually good questions. So, I'm going to play you guys a video from To Catch a Smuggler. The reason why I want to show you guys this clip of To Catch a Smuggler is I want us to think about some questions that we often ask for qualitative analysis. So these questions are, first off, what questions are the officers asking when they find this white substance? And also, what ways do they go about confirming what that white substance is? So I'm going to play that video, and then we can discuss those questions. Pause that recording. Get the recording going. All right. So what were some key questions that the officers started asking once they noticed they had a white powder? Huh? Yeah, so they made some real key observations about it. They were saying, I'm looking at this, I noticed that this is supposed to be in a coffee bag, but from what I know, coffee is not white, it's supposed to be brown. So they're asking, okay, what is this then if it's not coffee? Yeah? So that's the key question they were asking about it. How did they go about confirming what the white substance was? They obviously had some ideas about what it could be, because they said pretty quickly that it was which drug? Cocaine. What were some of the ways that they identified that? The smell, they looked at the texture, they noticed that it was white. The smell. I'm not sure what the smell of cocaine is, but I'm guessing it must have some sort of scent that you can recognize. So they had that question, but how did they actually confirm it? The little machinery in there. And then what did they do afterwards? They weighed it. So they wanted to see how much cocaine was there, and they also asked themselves if it was cut or not. So when they referred to that, they referred like, is it pure cocaine or have they added something else to it to basically lessen the concentration of it. So that's what they were doing. So what I want us to then think about when we are thinking about this is the difference between qualitative and quantitative analysis, because that is two different types of questions you are asking. Let me find some better pens because I don't like those ones for writing. And I gotta make sure I spell things properly because I am not a good speller. Where's my phone go? There it is. All right. Make sure I spell it right because there are two standards that we're doing that are relating: qualitative and now. There we go. Those are my two words. Okay. So the first one that we are thinking about is qualitative analysis. Oh, I don't like this pen much either. I miss my felt pens. I need to find them. I think I left them at home. So the first thing we are asking ourselves is qualitative analysis, and what is that? What word does qualitative kind of remind you of? Quality, good. So we're basically asking us when we have this question is, what is it? What is it? And that's often the first question that we are going to ask in a lot of scientific experiments, especially if you are thinking about like human body and trying to figure out like what is making someone sick and stuff. You ask yourself, what is it? And that's what they asked when they were doing their investigation on what the white substance was. And that was the same question I asked you guys when I showed you that blue Powerade, that we weren't sure if it was blue Powerade or if it was copper something. So we asked what it is. We obviously do questions to figure out what it is. You do things like identifying, classifying, you do flow charts. That's what you guys are going to be doing for this assessment is you're going to be given an unknown solution. You use the flow charts that you guys have been looking at for the precipitation reactions, and you identify what it is by process of elimination. So that is our first question, is what it is. This is very common, like I said. When I was doing my master's degree, the person that was also doing her master's at the same time, who happened to be American as well, we both did our projects in toxicology. My question I'll talk about later, but her question was basically, she was trying to understand why people that were taking a schizophrenic drug why were they having cardiovascular issues? So they were having heart problems, they were having heart attacks. So schizophrenics taking schizophrenic medication, they saw that there's a statistical increase in their chances of having a heart attack, and she was trying to understand why. So her first question for her thesis was, what is happening? So she looked at the drugs they were taking, and she was looking at what those drugs broke down to become, and was then investigating if those drugs that were broken down, did they cause the side effects and the effect to the heart, basically. So that was her question, was a qualitative analysis, or actually I should say her first question was a qualitative analysis question because she was asking what it was. She did eventually start asking the second type of question, which is qualitative analysis. Sorry, quantitative analysis. All right, so quantitative analysis. What word does that remind you of? Quantity. When you say quantity, what are you referring to? How much? So this question is asking, how much is there? So that's often our second question. So when she did her master's project, she asked what metabolites from the schizophrenic drugs are causing the heart problems, and how much is there? How much is actually being produced in the bloodstream that's now causing a problem? That's often a common question. And the reason why this question is going to be important is that if you guys are aiming for excellence on this internal, let me backtrack actually. So the first let me backtrack again. All right. This internal is going to be assessed in two parts. The first part is going to be a qualitative analysis, and you are asking yourself, what is in my unknown solution? Once you figure out what is in your unknown solution, the second part of the assessment is to write a small little write-up on the ions that you have identified. And with that small write-up, you are talking about what are the human and environmental impacts of that ion. So you're talking about what side effects does it have to humans, what potential damage does it do to the ecosystem. So that's the essay bit. If you want to get an excellence, one of the things you need to ask yourself with your essay is this quantitative analysis. How much is there? So when you guys are writing your essays, you really need to think about, yes, this ion is present, but is it present at a high enough concentration to cause problems? So for E on the essay section, you need to write about, or you need to explain how concentration affects impact on humans and environment. All right? Are we okay with that so far? Okay. So some things to also keep in mind, and this is something that my toxicology professor and now and also supervisor, former supervisor, he had a really good bit of information and when it came to how to think about effect on things. So I really, really love toxicology. Initially, when I was in my undergraduate degree at university, I wanted to do cancer biology as my specialization. I was really interested in cancer cells. I was also really interested in the immune system. I took a couple of papers, or a couple of classes on pharmacology because I wanted to understand how drugs worked, because how are these cancer drugs working in the body? One of the things that I picked up was that I picked up a toxicology because I wanted to understand why were there so many side effects with the cancer therapeutic drugs. That one paper in my last semester of university changed the trajectory of what I wanted to research because I loved his course so much that I wanted that to be my specialization. I no longer wanted to learn about cancer anymore. I wanted to learn about toxicology and the side effects of drugs. One of the important things that he said to us in his lectures was that everything is toxic. It is just a matter of dose. So I'm going to write that down because I love that saying. Everything is toxic. It is just a matter of dose. And that's what you really need to be thinking about if you're trying to get excellence on this internal, is how is that concentration of the ions you identified, how is the effect going to range depending on how much there is in the sample. And we will actually do an internal on quantitative analysis as our next one. That's going to be the titration one. But you do get to start thinking about it in this standard. So yeah, everything's toxic. It is just a matter of dose. The example I like to use is water. So water itself can be toxic to you. It is toxic to you if, in the instance, that you drink water and you do not go to the bathroom. You will actually die from, I think it's hypoxia? Something else I'm thinking about. Anyway, you drink water, you don't go pee, you're going to burst your blood cells and your brain's going to swell. And you're going to die from that. Now, what was really sad that has happened is in the States, there was a radio broadcasting group. I think I might have told some of you guys about this. And they had this radio contest called Hold Your Wee for a Wee. So they were winning a Wii gaming system if they drank water and held their pee. And one of the contestants actually died from that contest. So, and the thing that was really frustrating about this whole thing is that people were calling in, like nurses, doctors, medical professionals, and they were saying, you need to stop this contest. That contestant is experiencing water toxicity because she was complaining about dizziness and headaches and things like that. And her brain was swelling. And then she went home and she died later that day. There was a lawsuit, and I think they did win in the end, even though she signed a waiver, like a liability waiver, saying that like she accepts any risks that happens with this event. They threw that out because they're like, no, that is very obviously a health risk that you were doing. That was not just them taking a risk. That was genuinely a bad idea. So I'm pretty sure that's how it kind of played out. But yeah, so I use that as an example because I want you to make sure that you remember that even things that you have at a safety like level that you need to survive, like water, if you overdo it, it's going to cause problems. And I know some of you guys have had a look at my master's thesis. I'll just plop it down to kind of show you guys. So what I did for my master's, because I think it's interesting to talk about when we're talking about the impact of quantitative analysis, is that I was looking at drug therapy, and they were for hepatitis B. And with hepatitis B, you often take drugs in multiple combinations to help suppress the virus. I was looking to see if it caused any mitochondrial damage. I was looking to see if it killed the mitochondria. I had multiple different endpoints that I was measuring. It's good to have multiple endpoints because then you can verify if you know that your results are true or not. So I looked at ATP levels. I don't know if you guys have learned about that in bio, but that's the energy currency. So I measured that. I wanted to see if that changed. I also measured DNA levels because we suspect that these drugs interfere with the DNA replication of the mitochondria. Fun fact, mitochondria have their own DNA, and fun fact, you get that from your mom. It used to be its own living organism that was then engulfed by another cell, and they formed a symbiotic relationship. I measured that. I measured cell death, and I think there was what is it? Glutathione was the other thing I measured, which is an antioxidant. So it's a measure of like cell damage. So I looked at that, and you can see I used it in different combinations. The theory was if I used all the drugs together, you would have worse reactions than if you just used one drug. You okay with that? All right. I just like to show you guys this because you can actually understand some of this now. I'm just trying to find some of my graphs. See, I have my methods. Here we go. All right. So you can see I measured the number of cells to see if they were dying and things like that. Trying to find which one I'm thinking about. All right, so this is me just giving them short-term exposure to the drugs. I think it was maybe just a couple hours. I have to double check. But we can see the measurements on like what does it do to their cell number and if their cell numbers die. This one over here, I was measuring multiple days. So I did this. This is my thesis. I exposed the cells for 15 days to the drug. And then I exposed them for 20 days, and I just did markers to see if there was any change in their... This is a year. This is a year.
Oh, okay.
Yeah. So this one, I was looking at cell count. So I was just looking to see if I was killing my cells or not. Now, when I did this experiment, one of the things that we talked about, my supervisor and I, is we talked a lot about concentration and what concentration of drug I was going to use to test it. What I did in my thesis was I tested 100 times what your body would actually have. So if you took these drugs orally and they measure the blood concentration of those drugs in your system, it has an amount. I did 100 times that because if at 100 times, if I had no effect on my cells and it didn't kill them at a high concentration, then the concentration we were using therapeutically was safe. Does that make sense?
Miss, what drug did you use?
I used a whole load of them. I was using drugs on the cells.
No, on yourself?
No, not on myself.
Oh, okay.
On a cell line. These are all cells that were growing in a lab.
Any mess?
No. No, it was these guys here. So this is the 4 drugs I tested. Cytosporine, I think, is an immunosuppressant from memory. So if you have a organ transplant, your body can reject that organ. So you have to suppress your immune system to prevent your body from doing that. So that means you're going to be more likely to get sick and stuff. But the other flip side of that is you're going to lose your organ. So a lot of people that have hepatitis B might need a liver transplant because their liver dies. So I looked at that one. I think these 3 here I think 2 of them are used to treat hepatitis B, and 1 of them is used to treat HIV, because HIV and hepatitis B are often, you find those 2 diseases together. Because a lot of people that have HIV and hepatitis B, they've gotten it from like intravenous drug use. So if you're injecting illegal drugs into your body, you're sharing needles, you're going to be transferring a lot of the viral diseases. So if you transfer HIV, you're very likely to also get hepatitis. So that's why I was looking at these 4 drugs. I then tried these drugs in different combinations because my theory was that maybe there's a certain combination that causes problems. So on their own, they are safe. They tested them on their own and they're safe, but maybe in practice when they're used together, they cause problems. And the reason why I got to this as my thesis is that with HIV treatment, this is true in HIV treatments. So when you have HIV, you have to take multiple drugs to make sure the HIV virus is suppressed, and some combination of HIV drugs make a toxic side effect. So I was trying to see if that was true for hepatitis B as well, because the drug for hepatitis B is similar to HIV. We good so far? I just figured this is interesting stuff. If you want me to shut up, I can shut up. All right. So what I found in my research was that there was no toxicity to it. There was no additional cell death. There was no change to ATP levels, no glutathione change, no mitochondria change, or mitochondrial DNA change. So what that told me in my thesis is that this combination is probably safe. So that was my research. That's what I hear about that. Cool? Interesting? Hopefully. All right, I want you guys to see where research can go. Just checking our time. Cool, we got till 25. So last thing I want us, second to last thing that I want us to do. Oh, I should have deleted the slide because I put this on the next one. All right, so one of the things that is important for us to think about when we are doing water testing is the life force of the water and protecting that life force as well. We often think in a very westernized version of science, and we think about like us as being above the rest of everything else. Whereas if you look at other societies, in particular indigenous societies, they've included themselves in the circle of life and things like that, and they understand that we are a piece of the puzzle. They also recognize that, that definition of living and non-living is different than our definition of it. Or when I say our, I say Western definition of it. So if you talk to a lot of indigenous cultures like Māori, they do believe that things like water and the land have an energy force, have a life force, and are living in their definitions. So one of the things we got to think about is the importance of protecting water. And one of the ways that we protect our water is by identifying what's in it. So I have 2 videos for us to watch, just to kind of understand that importance. And then I have a scientist for you guys to research. Hopefully you guys aren't finding this too boring. I know it's a lot of talking. All right, team. What did we learn about Kepha Morgan when we looked him up? Yes. Several? He's a civil engineer, so he's trained as an engineer. What else? He's been an engineer for a very, very long time. What is his area of interest? Mostly water. He's very interested in water and water quality. What's an important background of his? He's Māori. He's indigenous. And that makes him view science in a different way. He is connected. Yeah. So he's more connected to it. He values water a lot more than what other people might value water at. And he's also bringing his indigenous knowledge to his field. So, something I want to talk to you guys about to keep in mind about science. I think I told this to my year 11s earlier, is that sadly, the scientific field is very white and male-dominated. And because it's white and male-dominated, can we pause our pack up? Important thing. Because it's very white and male-dominated, it's going to then direct the research in a certain direction. So a lot of the questions that they're going to be asking is going to benefit, males and white people at a subconscious level. So it's really important that we get diversity into science and into STEM because they're going to bring different knowledge to the table and they're going to ask different questions. They're going to ask questions that are important for their communities. So it's really important that we get more Māori and Pacifica people in, one, because they have a different set of knowledge, like his, about the life force of water, but 2, he's going to be asking the questions that are important for his community. So, important for his iwi, important for Māori, and then same thing, Pacifica, like, important about your health, stuff like that. So we need diversity in science. Okay?